Two. I was thinking about this. The first one was back in the 1980s. Yeah. So for years, my academic grandfather, Ed Lewis, who won the Nobel Prize, by the way, discovered and worked on what's called the bithorax complex. And that bithorax complex controls the identity of structures in the posterior part of the fly. So you can turn these structures called halteres into wings, and you can put legs on the abdomen and so on. I was working doing genetics, developmental biology on flies, and I discovered that there is another complex that we call the antennapedia complex that controls segmental identity in the front half of the fly. And one of the most prominent members of that complex is a gene called antennapedia, in which the antennae on the head of the fly are transformed into legs. There's another gene called proboscipedia, in which the proboscis, the mouth parts of the fly, are turned into legs. And I remember the day in the lab. I was all by myself, and I did a cross. I scored the flies, and it suddenly dawned on me that there was a cluster of genes just like the bithorax complex that worked in the anterior part of the animal. And I got very excited and started leaping around, and I called a colleague, Rob Donnell. We were graduate students together, and we proposed to do a big genetic analysis of the antennapedia complex. So that was the first thing. You get few of those aha moments in your scientific career. And when you get one of those, oh, you should cherish it. I mean, it was like drugs. I mean, it was, I was so excited. And then the other thing was, as a consequence of that, we cloned the genes in the antennapedia complex. And in the analysis of those genes, we discovered that there was a DNA sequence in the protein coding parts of the homeotic genes in the antennapedia complex, amongst all of the homeotic genes in the antennapedia complex and the bithorax complex. And that discovery was also made by some postdocs in Walter Gering's lab in Basel. And we made the discovery at about the same time. That was pretty exciting too, because we also found that sequence is conserved throughout all of the animal kingdom. And up until then, people thought that evolution was the invention of new gene. But it turns out that evolution is not the invention of new gene. What it is the reorchestration of the same set of genes that must have evolved 500 million years ago. It caused everybody to rethink the process of evolution. Homeobox is what that sequence is called. Caused a rather significant evolution in our understanding of evolution. Those are the two things. Discovery of the antennapedia complex and the discovery of the bithorax complex, or the homeobox. I remember the day when the postdoc who was doing that work came into the lab with the southern blot. People don't do southern blots anymore. And I saw the signal on there, and we just said, wow, because we knew it was in the coding parts of the genes. I was very lucky. There are things that contribute to being successful in science. You have to work hard, do lots of experiments, and you have to have a certain amount of luck. When I first started working on homeotic mutations in flies, I had no idea that you and I have these same genes. That was a big surprise to me. I went into it because I thought that homeotic mutations were strange and wonderful in and of themselves. I didn't do it because I thought I was going to discover something important about evolution. Maybe insects, maybe arthropods, but mammals? No. To get the best science, you have to work hard, you have to really go through the literature, and you have to have good colleagues, and you have to have good collaborators. And you can't do it all yourself. The days of, Isaac Newton laboring away in the countryside but not all of us are that brilliant, right? And so it's always good to have good colleagues and good students. Science is a collaborative effort, especially nowadays, because you can't do everything. The technology is complicated nowadays. You need to have engineers, you need to have chemists, you need to have, importantly, bioinformatics has become very, very important. Microscopy is incredibly important because now with all the single cell gene expression, what you need to do is you need to see where all those genes are being expressed. You need tag things, you need to find out what organs, what subcellular domains are these proteins being expressed in. And the best way to do that, of course, is to look and see what's going on. And then also what you need microscopy for is you need to look at the morphological consequences of altering that gene expression. And the way you do that is to look and see what changes in organogenesis and morphology of cells. All of these things are important. What I try to tell the students is that you should have a good question, a good biological question, but you should not limit yourself to one piece of technology. You should bring everything you can find to bear on answering that question. And that's why you have colleagues, because there are other people down the hall in another lab who are very good at something you're not very good at. And so you go and you talk to them. And you get them to help with the research project if you can get them to be interested in that project. Learn as much as you can. The human brain has got so much capacity, right? You can load as much as you can into your hard drive, but then, there are only so many petabytes of information you can stick in there. It's good to have somebody else work with you. Bioinformatics is incredibly important. That didn't exist when I was going to graduate school. There was no such thing. People didn't have computers in their lab when I was in graduate school. I mean, I remember typing up my thesis on a typewriter. Who has a typewriter anymore? One thing you have to have, and this can degrade more so in this country than I think in Europe, is the support of the administration of the university and the institution. Here in the United States, universities have an important component that doesn't exist where you live, and it's called athletic. So we have football and basketball. That's a money-making scheme for the university, and they put a lot of time and effort into that, and sometimes it detracts from the institution's support of basic research. Places like Harvard and Stanford, where they're private schools and they have independent sources of wealth, but state institutions like where I work, it can be a problem. So it's a constant battle to get support from the institutional administration to make sure that they maintain the infrastructure for doing science. It shouldn't have to be the fight that it is. So I've tried to avoid the administration as much as I possibly can, because their interests are going to cocktail parties and things like that, and I have no interest in that. It's very important to realize is that different fields doing molecular biology and genetic, and I can speak to flies. When you do work on flies, what you have to do is you have to create a whole series of reagents. None of my ecology colleagues have to do any of that. And their science tends to be somewhat observational and correlative. Many of them are less involved in, and frankly, just don't care about underlying mechanisms. Correlation is good enough for them, and that's where their analysis stops. We can't do that in molecular biology. You could never publish a paper in a reputable journal that was just correlation. You have to have data that address mechanism. So, that being the case, you have to weigh quality and quantity depending on the field. One paper that has required that the person develop a whole series of reagents to do the experiment is very different than a paper where somebody went out and counted something in the wild and said, oh, okay, this bird did something this many times, and that means blah. Why the bird did it that way doesn't matter, okay? But that publishing that in a reputable journal. You can publish a lot more papers in some fields than you can in others. So you have to balance that out. And so you have to consider a lab with a small lab in molecular biology. If they publish 4 papers in a year of high quality, where they have gotten all of the necessary reagents, that's different than somebody in a correlative field where they've published 40 papers. So that has to be taken into consideration. And what you need to do is, you can't just stamp a number on it and say, you have to publish 20 papers a year. Now, there are some labs that I know of, people that I collaborate with, where they have 40 postdocs in a lab, at a institution like the Harvard Medical. And yeah, they can publish 20 to 40 papers a year in high quality journals, but that's because they have the Roman army to to do the work, right? Yeah, they can go conquer the Gauls. So but a normal lab with a normal number of students, a normal number of technicians, you have to consider what the nature of their research is. To us as scientists, it's obvious. Think of all of the things, okay, let's do a specific example. RNA vaccines. Has that been helpful? Well, piccolo, huh? And there's just innumerable things. Basic research, who would have thought that working on viruses that kill bacteria would ever have had the enormous impact? I mean, think of cloning. Nobody even thinks about cloning genome fragments anymore. It just happens. Sequencing DNA. Look at the technology and all of the things that have come out of that. All of the GWAS studies that have been done to find disease causing. So some poor person, sick person, presents in a clinical environment. They have no idea what's going on with this poor person, but they look and they say, gee, this person has relatives that have similar problems. They sequence the person's DNA. They then, which can be done now, if you want to have your genome sequenced, you can go have it done. And it's affordable. Who would have said that even 10 years ago? Even some ability to, I wouldn't say cure a genetic disease, but at least ameliorate the problem. I have a friend named Ross Kagan, who is now in Scotland, is interested in cancer. And he uses Drosophila as a way to study cancers. And he can create fly lines, that either by making mutations in the fly's genome or by putting in human disease associated genes, and he can make the fly sick. Usually what he does is he makes eye defects, because the fly can survive without an eye. And then he takes those flies and he uses libraries of small molecules to see if he can cure those defects. And he finds small molecules. And then they go on and do clinical studies to find out which of those molecules will not make the patient sick all by themselves and which will ameliorate. And he's found a couple of drugs that way. I mean, think of all of the technology that's available to everybody. Think about these things. It just didn't fall out of the sky. So, basic research, you need to support it, because you don't know which basic research is going to have the long-term effects. Like I said about homeotic mutations and Hox genes. I had no idea that they had the far-reaching effects on evolution, and it turns out there are Hox genes that are associated with chromosomal rearrangements that are associated with cancers. You just have to support basic research and realize that some of it is going to be important and will produce important results. others maybe not. We live in an age where politically people are rejecting science. I was in school when Sputnik happened. The people freaked out. The Russians are going to take over the world because they launched this ball with an antennae coming out of it. And what it could do is it could beep. That was it. But they had the rocket technology to make this beeper thing to go like a cat toy, right? So what happened when that happened was society freaked out. Money was poured into basic research like you can't believe. Okay. Back in those days, NIH grants were scored on a 1 to 5 basis, 1 being good, 5 being not so. So it used to be, and it is now, that you have to get a 1 to get funded. But back then, they poured so much money in it, you could get a 5 and still get funded. The only way you wouldn't get funded is if you got a not recommended for funding. So if there was the smallest amount of scientific merit in the proposal, it'd get funded. Okay. Well, that doesn't happen anymore. So do you think so we just had this whole COVID-19 disaster that took place. And now monkeypox. What has been the result? Are people freaking out? No. People are saying, I don't want to get that vaccine. Are we pouring more money into understanding how to use RNA technology and DNA technology to make better vaccines more rapidly? No, we're rejecting the paradigm. And when I say we, I mean society as a whole. I don't know if it's this pazzo in Italy as it is here, but here I'm old. I'm not going to live to see the worst of it. But I feel very badly for people, young people like you and my daughter, who are going to have to live through that. And to see the politicians not be willing to do anything, it's depressing.